In tonight's lecture, we're going to review Chapter 1, An Introduction to Biology. Biology can be defined as the scientific study of life. But what is it that distinguishes living things from non-living things? The phenomenon of life seems to defy a simple, one-sentence definition, yet almost any child perceives that a dog or a bug or a plant is alive while a rock is not. We recognize life largely by what living things do. So we will first discuss the properties of life that are similar to all organisms. The first property of life we will discuss is order. All living things exhibit a complex but ordered organization. And we can see here on the PowerPoint slide that starting with the smallest unit of matter, the atom, and we can build upon that into a complex system called an organism. And we will discuss this more as the lecture progresses. Also, all organisms respond to stimuli. So in other words, all organisms have the ability to respond to the external environment. Moving on to the next slide, organisms also have the ability to reproduce. There's a little bit of gray area associated with this property of life. For instance, mules are often sterile. If something is sterile, it means that it cannot produce offspring. But nonetheless, a mule would certainly be considered an organism. All organisms also have the ability to grow and develop. Information carried by genes control the pattern of growth and development in all organisms. All organisms also have the ability to regulate. In other words, the environment outside of an organism may change drastically, but the organism can adjust its internal environment, and that helps keep it within the appropriate limits. Moving on, another word for this ability is homeostasis. So homeostasis is really simply the ability to maintain a stable internal condition. The human body has numerous homeostatic mechanisms that helps it to maintain the correct body temperature, pH, glucose levels, etc. Organisms also have the ability to utilize energy. In other words, all organisms take in energy and use it to perform all of life's activities. We see this in plants. The ability to capture solar energy and convert that into chemical energy is called photosynthesis. Cellular respiration is another form of energy usage, and in this case, when you consume food, the food is converted into energy that your body can use to do work. And this brings us to evolution. Reproduction underlies the capacity of a population to change or evolve over time. Remember, evolution only occurs on a population-wide scale. So changes, mutations that happen in the DNA will allow organisms, or I should say could allow organisms to be better suited to their environments. And that brings us to the levels of organization of living things. So looking at this slide, we can see that the atom is the smallest unit of matter. And then a molecule is two or more atoms that are bonded together. So simply put, molecules are clusters of atoms. And then macromolecules, well, these are large molecules, and there's 4 macromolecules that you need to be familiar with. That's DNA, proteins, lipids, and carbohydrates. And then the molecules lead us into the organelle level of organization. Organelles are functional components of cells, such as the nucleus that houses the DNA. And from organelles, we're led into the cellular level of organization, and we will define the cell as the smallest unit of life. In other words, the cell is the smallest unit that can display all the characteristics of life. And cells will combine together to form tissues. A tissue consists of a small group of similar cells performing a specific function. For instance, your cardiac muscle is an example of a tissue. And from tissues, we're led into the organ level of organization. So an organ is just a functional structure that's made up of tissues. For instance, your heart is an organ, your liver is an organ, and these organs are made up of groups of tissues. And from there we find the organ system level of organization. So an organism's body consists of several organ systems, each of which contains 2 or more organs. For example, your circulatory system consists of your heart and blood vessels. And that leads us into the organism level of organization. An organism is a living individual thing, like a person, like a dog, like a cat, like an iguana, or it can be a single-celled prokaryotic species such as E. coli. And then we move into some larger organizational levels of life. And your PowerPoint is defining this as the biosphere level. But in the biosphere level, we can break this down into the population. So the population is a community, which are groups of interacting individuals of one species, such as a group of people or a group of iguanas. And that leads us into the community level of organization. And that is all of the populations of species living in a specific area. For instance, all organisms in a tidal pool, which would include iguanas, crabs, seaweed, bacteria, and other organisms, are collectively called a community. And then we move into the ecosystem level of organization. So as I referenced a tide pool, like for instance a tide pool in the Galapagos Islands is an example of an ecosystem. An ecosystem consists of all organisms living in a particular area, as well as the non-living physical components of the environment that affect the organism, such as the water, air, soil, and sunlight. And that leads us to the biosphere. The Earth's biosphere includes all the environments on Earth that support life. Moving on to the diversity of life. Diversity is a hallmark of life. The pie diagrams on the slide show that scientists have discovered approximately 1.8 million species of organisms throughout our Earth's biosphere. This diversity of life includes about 290,000 species of plants, about 59,000 species of vertebrates, in other words, animals with backbones, and about 1.3 million species of invertebrates, or animals without backbones. And in this next slide, we're looking at a phylogenetic tree. So a phylogenetic tree is simply a diagram that shows the hypothesized evolutionary relationships among organisms. And this brings us to taxonomy. Taxonomy is the classification of organisms based on similarities. Carl Linnaeus, a Swedish botanist, is largely credited with inventing the modern system of naming organisms. So taxonomy is really just the scientific study of naming. So by examining this slide, we can see that there are 3 domains of life: bacteria, archaea, and eukaryota. And then we can further break organisms down into their kingdom, phylum, class, order, family, genus, and species. So binomial nomenclature is where we call an organism by its genus and species. In other words, Homo sapiens, Dalbergia stevensoni, Escherichia coli. That's an example of binomial nomenclature. And in this next slide, we just see a pictorial representation of the taxonomy that we've been talking about. So in the upper right corner, we see the domesticated dog, and how when we move from the species level to the genus level, that the domesticated dog is related to the wolf. And when we move from the genus level to the family level, we see the domesticated dog, the wolf, and the fox, etc., all the way up. And if we look down in the lower left corner, when we look at domain Eukaryota, we see that all of these organisms in this eukaryotic domain include the domesticated dog. Well, we see the lion, we see the tree, the mushrooms, the protist. So the gist of it is, at the domain level, you're related to more organisms than you are at the species or the genus level. And this brings us into scientific method. Recall the first definition from the start of this chapter. Biology is the scientific study of life. Now that we have explored the question of what life is, we can turn our attention to the next obvious question: what is science? And how do we tell the difference between science and other ways of trying to make sense of nature? The word science is derived from a Latin verb meaning to know. Therefore, science is a way of knowing, one that is based on inquiry. It developed from people's curiosity about themselves and the world around them. This basic human drive to understand is manifest in two main scientific approaches. One is discovery science. In this science, it's mostly about describing nature, and the other is hypothesis-driven science, which is mostly about explaining nature. Most scientists practice a combination of these two forms of inquiry. In discovery science, scientists seek natural causes for natural phenomenon. This limits the scope of science to the study of structures and processes that we can observe and measure either directly or indirectly with the help of tools such as microscopes. This dependence on observations that other people can confirm demystifies nature and distinguishes science from belief in the supernatural. Science can neither prove nor disprove that angels, ghosts, deities, or spirits, whether benevolent or evil, cause storms, rainbows, illnesses, or cures, for such explanations are out of the bounds of science. Verifiable observations and measurements are the data of discovery science. In our quest to describe nature accurately, we discover its structure. In biology, discovery science enables us to describe life at its many levels, from the ecosystem down to cells and molecules. Darwin's careful description of diverse plants and animals he collected in South America is an example of discovery science. A more recent example is the sequencing of the human genome. Discovery science can lead to important conclusions based on a type of logic called inductive reasoning. An inductive conclusion is a generalization that summarizes a large number of observations. All organisms are made of cells, as an example. That induction was based on 2 centuries of biologists discovering cells in every biological specimen they observed with microscopes. The careful observations of discovery science and the inductive conclusions they sometimes produce are fundamental to our understanding of nature. Moving on to hypothesis-driven science, the observation of discovery science stimulates us to ask questions and seek explanations. Ideally, such investigation makes use of what is called the scientific method. As a formal process of inquiry, the scientific method consists of a series of steps. These steps guide scientific investigations, but working scientists typically do not follow them rigidly. Different scientists proceed through the scientific method in different ways. Most modern scientific investigations can be described as hypothesis-driven science. A hypothesis is a proposed explanation for a set of observations, an idea on trial. We all use hypotheses in solving everyday problems. Let's say, for example, that your flashlight fails during a campout. That's an observation. The question is obvious. Why doesn't the flashlight work? A reasonable hypothesis based on past experience is that the batteries in the flashlight are dead. Once a hypothesis is formed, an investigator can use deductive logic to test it. Deduction contrasts with induction, which, remember, is reasoning from a set of specific observations to reach a general conclusion. In deduction, the reasoning flows in the opposite direction, from the general to the specific. From general premises, we extrapolate to the specific results we should expect if the premises are true. For example, if all organisms are made of cells, premise 1, and humans are organism, premise 2, the humans are composed of cells, deductive prediction about a specific case. In the process of science, the deduction usually takes the form of predictions about what experimental results or observations we should expect if a particular hypothesis or premise is correct. We then test the hypothesis by performing an experiment to see whether or not the results are as predicted. This deductive testing takes the form of if-then logic. So we make an observation. My flashlight doesn't work. Then we ask the question, what's wrong with my flashlight? Why doesn't it work? From there, we formulate a hypothesis. The flashlight's batteries are dead. And then we make a prediction. In other words, we say, if I replace the batteries, the flashlight will work. Then we do the experiment. We replace the flashlight's batteries with new ones. And then one of two outcomes. The experiment supports the hypothesis. In other words, it makes the prediction come true. We were right. When I replaced the batteries in the flashlight began to work again. Therefore, the batteries in the flashlight were dead. Or it could also happen like this, where the hypothesis is not supported. In other words, we replace the flashlight's batteries with brand new batteries, and the experiment still doesn't work. So it does not support the hypothesis. Then we go back to form another hypothesis. Well, the flashlight bulb is burned out, or the batteries that I bought are defective. And from there, we begin the deductive process once again. However, in some experiments, there's more at stake than the flashlight's batteries running out at your camping trip. In other words, when we get into the medical profession and when life is at stake, we need to have more concrete ways of being able to support or reject a hypothesis. So these experiments will often have what we call a control group. A control is a part of the experiment that does not change. A control is a point of comparison in which other test results are compared. So this slide is really just basically illustrating for you a controlled experiment. The experimental group is the group of patients who will get the blood pressure treatment, and the negative control group is the group of patients who get no treatment. They get a placebo or a sugar pill, but they don't know they're getting that. That's key. If they knew they were getting it, there may be some sort of psychological effect, and we don't want that. We want our deduction to be true and clear, so we use the negative control group as a basis for comparison against our experimental control group. And in this process, we collect data. So we will glean data from both the experimental and the negative control group, and then we will be able to analyze those results and compare back to the hypothesis. These results then help scientists to deduce a scientific conclusion. So moving on to the next slide, remember the results, if they support the hypothesis, we don't prove the hypothesis in that respect. We simply make a new experiment that will be based on the results. And if the experiment does not support the hypothesis, then we go back to the drawing board and try again. And you can, once again, look at the flow diagram on the right. So remember, the key point here is that hypothesis is never proven true. It is only supported. And this brings us to reporting scientific work. Scientists often share their findings for their research with others, and this helps them to expand upon their body of knowledge. It helps to advance science. One way that scientists will do this is through peer-reviewed articles. And in this final slide, we have a quick review. These questions should help you to reflect on tonight's lecture.